Hello, my name is D'Artagnan Sloan, and today I'm going to be presenting on the pigment section of the biochemistry unit. So, all pigment molecules have intense absorption bands in the visible region of the spectrum. The color is not what is absorbed, but instead what is reflected. So these structures are going to be highly conjugated. Absorption and reflection are dependent on the color wheel, which I will explain in one moment. Absorption and emission spectra can be used to determine what is absorbed and what is reflected, and the part of the molecule responsible for absorbing the radiation is called the chromophore. So the color wheel that you see in the bottom right hand corner is going to be how we tell what is reflected, and the complementary color, which is the color opposite of the color that is absorbed, is going to be what is reflected. So for example, if purple was absorbed, then yellow is going to be what's reflected. Next, we're going to talk about porphyrins. These contain a planar ring containing 4 heterocyclic rings, all containing carbon and nitrogen, and are linked by bridging carbon atoms. It acts as a ligand, forming chelate with metals that have coordinate bonds. Different porphyrin compounds contain different metals and also differ in the nature of the substituent groups that are attached to the carbon atoms. Chlorophyll is the primary photosynthetic pigment found in plants. Although different forms of chlorophyll exist, they all contain magnesium, which is key. So they absorb light strongly in the blue part of the region of the spectrum and lesser in the red, which gives it that green color. Accessory pigment, they are examples of accessory pigments that harvest light in different parts of the spectrum and pass their energy to the chlorophyll. So the chlorophyll undergoes a redox change where electrons are passed to electron transport carriers, and then the chlorophyll then returns it to its original state by gaining electrons from water. So here is an example of chlorophyll absorbing photons from light energy, and then electrons are transferred from the chlorophyll to an electron transport chain, and then it stores the energy or stores it as energy. The thermal stability of chlorophyll depends on pH and a few other. It depends on pH. Magnesium is lost in the acidic solutions and replaced by 2 hydronium ions. And cooking food also breaks down cell membranes, which releases acids, and that in turn decreases the pH and brings about color change from green to an olive brown. Chlorophyll is going to be more stable in alkaline solutions because of the stability depending on pH. Hemoglobin and myoglobin, called the oxygen-carrying team. Hemi is a prosthetic group found within the protein molecules. Hemoglobin is going to have 4 hemi groups, each bound with a polypeptide chain with a quaternary structure, whereas myoglobin is going to have 1 hemi group and a single polypeptide chain. Hemoglobin carries oxygen and the myoglobin stores it. So both molecules bind reversibly with oxygen, which forms a weak oxygenated bond with the iron. The compounds are then known as oxyhemoglobin and oxymyoglobin. The reversibility of this is going to be essential because it enables the oxygen to enter the lungs and be converted into oxyhemoglobin, then leave and be converted back into the respiring cells. So the uptake of oxygen by the second, third, and fourth hemigroups within a molecule gets easier as oxygen binds. Basically what that means is that as oxygen binds to the structure, to the different hemis, it's going to increase the oxygen affinity. So each time an oxygen bonds, it'll eventually become easier for another oxygen to bond. From the graph, we know that at low concentrations of oxygen, hemoglobin has a low affinity for oxygen, and at higher concentrations of oxygen, the opposite occurs. In example I, we can see the reversibility of the hemoglobin and oxygen to oxyhemoglobin reaction. And so this causes uptake where oxygen concentration is high and shifts to the left in respiring cells, releasing oxygen where oxygen concentration is low. And then other factors that influence the binding of oxygen and hemoglobin are temperature, pH, and carbon dioxide. So increasing temperature reduces the affinity for hemoglobin for oxygen as the dissociation curve shifts right, and then the decreasing of pH reduces the affinity for hemoglobin as well. And so does the introduction of carbon dioxide, because an increase in carbon dioxide will reduce the affinity for oxygen. And so here we see some examples of how different reactions and different conditions shifts the curve. Specifically focusing on the percentage of carbon dioxide, hydronium, which is pH and temperature. And then here is the forward reaction for hemoglobin and oxygen, forming that oxyhemoglobin. This is going to take place in the lungs, where it's high oxygen percentage and lower temperature, and high pH with low percentage carbon dioxide. And then the reverse reaction is going to occur in respiring cells, where it's going to be low percentage of oxygen and a higher temperature with low pH and a higher percentage of carbon dioxide. The 4 polypeptide chains in adult hemoglobin are going to be 2 alpha chains and 2 beta chains, whereas in a fetus it's going to be 2 alpha chains and 2 gamma chains. The fetal gamma chain is going to have a greater affinity for oxygen, because it has to pull the oxygen from the maternal blood, because the fetus doesn't actually breathe, so it can't get oxygen other than from the maternal blood. And then myoglobin is also going to have an oxygen dissociation curve to the left of hemoglobin. It has to have a higher affinity for oxygen as well, so that it can actually take the oxygen from the hemoglobin and store it. Without this higher affinity, it wouldn't be able to store it and actually take the oxygen from the hemoglobin. So the myoglobin would just be useless. Carbon monoxide is a toxic gas that's harmful to humans because its affinity for hemoglobin is 200 times higher than oxygen. This is especially dangerous because with the presence of carbon monoxide, it'll bond to the hemoglobin instead of the oxygen, meaning that you won't be breathing in oxygen, you'll be breathing in carbon monoxide instead. And this causes carbon monoxide poisoning, which will lead to death and choking. Next, we're going to talk about cytochromes, which are electron transport carriers. This is a varied group of protein molecules that also contain the hemiprosthetic group, found in membranes, and they are responsible for the electron transport during the redox reactions of aerobic respiration and photosynthesis. So this is going to be found in both animals and plants. During these reactions, the cytochromes are reduced and then reoxidized as they accept and pass on electrons. They are organized in sequence by their electrode potentials so that the electrons flow down an electrochemical gradient. In cytochromes, the iron of the Hemi group changes its oxidation state between +2 and +3, and the final cytochrome in the process passes its electron to the oxygen with the formation of water. This is actually where the poison cyanide occurs, because it prevents that last step. Carotenoids are a group of pigments containing long hydrocarbons with many double bonds. They range in color from yellow to red and are fat soluble due to the long nonpolar hydrocarbon chain. The carotenoids are found in plant leaves and help in the harvesting of light for photosynthesis. They're the example of accessory pigments that I mentioned earlier, and they help to pass light energy to the chlorophyll. So they will absorb the light energy and then they pass to the chlorophyll to be used. The double bonds between the carbon atoms that give the carotenoids their color also make them susceptible to oxidation. This is why they act as antioxidants. The oxidation can lead to a loss of vitamin A activity as well as a loss of color and give off odors due to the release of volatile compounds. Not much is known about the oxidation of carotenoids. However, we do know that it involves the conversion of trans carotenoids to their cis isomers, and this can be reduced by preventing exposure to air and light and by decreasing their storage time. So we do not want the oxidation of carotenoids because it reduces their value and their health. Anthocyanins are responsible for many of the pink, red, and blue colors of plants. They absorb strongly in the blue and green parts of the region, which is why the more red colors are going to be reflected. They're formed by a reaction between sugars and proteins that requires light, and the color of anthocyanins also changes as the pH of cell sap changes. So generally pink in acidic conditions, purple and neutral, and greenish yellow in alkaline conditions, which is why, due to their sensitivity, they can be used as pH indicators. So we would see pink in acidic conditions, purple in neutral solutions, and then greenish yellow in alkaline solutions. And this arises from the transfer of H+ ions from OH groups, which alters the conjugation and absorbance at the chromophore. And these are going to form deeply colored coordination complexes with Fe3+ and Al3+ ions. And down below, you're just going to see some examples of some anthocyanins. And so finally, we're going to analyze pigments. So there are two ways to do this. One of them is going to be paper chromatography. It's described in depth on page 701, but basically you would separate the different pigments through their differences in solubility by having them displayed on a piece of paper. And then the other is going to be thin layer chromatography, which is an example of adsorption chromatography. It's a stationary phase is a thin layer of adsorbent particles of alumina or silica, which may only be 0.2 millimeters thick. It's going to be supported on a glass or plastic plate, and the mobile phase is going to be the solvent, chosen according to the chemical nature of the pigments. So the mobile phase can be different for each pigment that's being analyzed. And then the pigment extract is placed on the origin, and capillary action causes the different compounds in the spot to separate. So generally, thin layer chromatography is going to be quicker than paper chromatography, and it's going to work with smaller samples. So it's used more often just because it's better and more useful. On the left, you're going to see an example of paper chromatography, where the paper is going to be submerged in the solvent, and then the pigments are displayed on the paper. And then on the right, it's going to be an example of thin layer chromatography, where the mobile phase is going to be the solvent, which is going to depend on the different pigments, and then the pigments are displayed as well. Thank you for watching my presentation, and that is all on pigments.